Sure, let's just keep going, see what else we see before we go up shallower. Kind of hang here a little more.
There are those like gelatinous tubes in
We were just starting to
Argus Cam. They look like tubes with like little strings of like orange beads in them.
snuggie on the wall over here.
Hold on.
There's a
I'll point one out
lot
if I
in
see it.
Argus Cam.
Yeah, there's a lot going on.
Can
But
I,
they're like kind of like slowly moving through. Maybe they're salps?
Yeah, it's one thing that came to my mind.
They move kind of fast though.
Oh, really?
Like, not super fast, but like controlled movement.
Hmm.
Oh, there's one.
There's another snapper.
Oh. Oh, they keep disappearing.
Are you wishing you had a button?
I mean, even if I got a screen grab, it would be nice to be able to like share it. But yeah, there's these like little creatures that keep popping in and out of Argus view, and I'm trying to figure out what they are.
I wonder what the stuff we picked up was.
The brown coral?
Yeah, because it's like everywhere.
Yeah,
I think it is a coral based on the way it was growing.
It's definitely everywhere.
Yeah.
So Hannah, we have a question coming in from a viewer about, would the light causing the swarm of invertebrates around Argus and Herc make the transect have an inflated species count by possibly attracting more fish?
Can you repeat that again? I had some background noise.
Would the light causing the swarm of invertebrates around Argus and Herc make the transect have an inflated species count because of maybe them being attracted to all the possible food?
Yeah, it definitely could influence. Definitely. Although I wouldn't say like for all the fish species, maybe not so much. Not for all of them. I mean, maybe some, or they're curious and checking it out. But that being said, Herc is pretty loud, so at the same token, might be skewing the survey because it's scaring other fish away that won't come closer. Yeah, so it's definitely
double-edged sword.
yeah. Yeah, one really cool thing about closed circuit scuba diving, when you're using rebreathers, and when you're using those, you can go to these mesophotic depths. And one really cool thing is because it's closed, you don't have a closed circuit, you don't have air bubbles coming out, so you don't have the noise. So doing fish surveys, you can just be like another fish in the water. You're silent, right? I have a friend, she did it, and she said one of the coolest things is you can just kind of sit on the bottom floor, just kind of float and bob over a reef edge and just stare at everything coming, and things come so much closer because there's no noise, because the noise scares them away, obviously.
Oh, there's a little jellyfish in Argus Cam.
Where is he at?
Right in the center.
Oh, he's tiny. Is that a jelly?
Maybe it's a ctenophore. I thought I saw like 4 tentacles.
Oh, yeah, it kind of looks like it.
Hello.
Oh, another trevally. Is that another one or the same one?
I don't know. It's pretty bold to chill out by ROV.
Yeah, he's
There he is
really
again.
curious. Interesting, they're coming so close. Whenever you're diving, I feel like you never really get too close to them. Could we sample this whip in front of us? Or another one if the boat's moving too much right now? I would like to get one at some point. Like I said, it doesn't have to be now if it's not feasible. It's a tall one. Do you want to get some stills of that, Peyton, before we sample?
Sorry, what?
We're going to sample this
Oh,
sea
yes,
whip.
yes,
If
yes.
you want to
I
just
do want to get some
get
crabs.
some grabs, yeah.
Can we get a close-up soon?
Oh, okay. You want to try to put it in a suction sampler?
Sounds good.
Okay.
They're all open, right? Yeah. Or did
one
we
is
use one?
one has something in it.
Okay.
Could we get a? Actually, no, it's fine.
Oh, did you want to close it first?
I can wait until after it's cut, because then it'll be in the same place.
Oh, look at that.
You get it?
Did you get it?
No.
Well,
Oh, is it gonna fold? It's getting tricky.
Sure.
116.7. Thank you.
Bouncing everywhere.
We do have a request for Gorgonians. We'll have to get some more.
And zoom in.
Yes.
What was 54?
This is sample 54.
I haven't got it yet.
Dun.
Oh, I think those things are salps.
Why is the
Oh, did you
jaw
see him again?
drift?
Argus
I
cam?
saw one drifting by in front of Herc, actually.
Oh. It looks blurry. I wonder if there's a mix of...
Like water mixing?
Yeah.
Yeah, I noted
Like-
some
I don't like
shimmering
the
earlier too.
wrist
I'll,
tape drifting like that?
Yeah.
Could you take a Niskin bottle to figure that out?
We could test salinity.
So check it out. Ready?
Yeah, it's
Not on
interesting.
those.
Maybe that's a good idea. I would take a Niskin sample to see what's going on with the water mixing.
I don't know if it would tell us much about mixing per se, because it would just homogenize.
Oh, this is true.
But
Yeah.
I guess I was thinking we're too deep if there's like a freshwater.
can we get a
input.
zoom of it before we slurp it?
You want to do what?
Oh,
Little.
What was that?
Could I get a
Is-
zoom on it so I could get a capture before we slurp it? Thank you. Sorry.
It's okay.
oh, I think that is a salp. I see it.
How's that?
I think that's what Peyton had seen earlier.
Great. Thank you guys.
Yeah.
Sweet. Thank you, thank you.
Slurp jar 2.
is going. Doesn't look like it's being sucked in at all.
It started to for a sec.
Oh, there you go.
There you go.
There it goes. Dun 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 dun.
Zoom wide.
Made it in yet?
Haven't seen it.
Yeah.
I don't think that's lined up. A little bit more forward.
There's definitely some little invertebrates inside of it.
Oh, too much. Yep, right there. Ooh.
Uh-oh.
Not sucking.
Well, we did get some of the small invertebrates.
That is definitely not lined up. All right, rotate forward. A little bit more.
Hey, Lily, have we reached TN4 waypoint? No, right? Yeah, if you don't mind zooming out.
0.
Yeah. The same spot.
4.
Oh, I see T4. Okay. So
Alright,
it's been
then what's going on
about
with our
2
jaws?
hours zigzagging. So yeah, maybe after this we move up a bit more so we
jars aren't
can make
rotating.
it to T4 by, I guess we have to make it there within the next 2 hours. So yeah, what's the
It's
distance
not going forward.
from our, meters? Oh, it's not that far. Straight. Okay. Yeah, maybe we. Okay.
Going off SPLs.
Yeah, so maybe we go. Yeah, like let's see. Maybe we go halfway between where we are into T4 and then we can zigzag laterally in there maybe a little bit before heading to the top. Let me see what the depth is at the very top. The very top is about 90 meters. Yeah.
You know the SPL, Lily?
It says it's 90.
We're going to take another clipping of this and put it in a bio box.
Okay, it never came through?
Just to be sure.
And our jars aren't lining up, and they stopped rotating.
Uh-oh. Okay.
I'm going to list this as 54 because we did get a few small invertebrates in slurp jar number 2.
55?
This next
Zoom in.
one will be 55.
How's that?
Good.
Man, that's kind of hard to...
It's so thin.
Yeah.
Oh.
Or-
Do you want the whole thing?
Didn't want the whole thing.
Come on. Oh, geez.
There you
There
go.
you go.
Nice.
I think that might fit in the
Whoa.
small bio box.
Small bio boxes would work, so that way we can save room for other large ones. Oh, are we doing front? That's
Sure,
fine.
starboard. It's so light, I don't know where it's going to float when I let go of it.
Oh, gotcha. We could do forward bio box B if you'd like.
Okay.
So the tool track. Ready?
Go ahead.
Perfect.
Nice. and
Great.
easy.
Awesome. Great job.
Okay, so just towards TN4 now, Hannah?
Yeah, let's move. There are these 100, or you said it was 120 meters?
It's 180 meters.
180 meters. So let's go like another 50 up,
Okay.
and then we'll start to
This thing is
zigzag back and forth.
Okay.
We can just kind of, how many knots have we been moving at? What's our speed been?
We're not really. So we go about between like 1.5 and 3 knots, but we're
Okay.
doing steps just because we were on a steep cliff.
Yeah, okay.
So 1.2 or so is what I would average.
Okay. Yeah, I guess we'll just slowly start working our way up. Unless if
Okay.
we see something of interest, then we'll start zigzagging laterally. If not, we'll go up, like I said, maybe about 50 and then start zigzagging. Whoa,
Okay.
that's a dense
Wow.
patch. Wow.
My goodness.
Yeah. Ooh.
Greetings, Birmingham, Alabama. And to answer the question that came in while we were sampling.
Okay, I had my mic off, so what's going on?
So we're heading just 50 meters or so towards the next waypoint, just kind of up, which is at 140. So it's just kind of up this slope right now.
Okay.
And then once we get there, or before if we see something of interest, then we'll start zigzagging laterally again.
So viewer who asked, we are all technically in the same room. We're in the control van on top of the Nautilus. So we have 2 rows, the front row and the back row. The back row is where science and data is, and then our front row, like here is where SciComm and video and Argus and Hercules and navigation all are.
If you're looking in the quad view on the right-hand side of what it's currently looking is nav going left. Yes, she's waving.
Yep.
And then going left in the middle is the Herc pilot, and then over here next to, there you go, is the Argus pilot. And then let me angle the camera some to on video, and next to me, you can't quite see him, but there's Brian. And turn on.
My
Reading all your
disembodied head.
questions. Yeah, the disembodied hand from around the corner. And then if I keep going around, there's the back row and all the science people. Peyton in the back and Hannah
We're
in the front.
frozen.
So that's the control van.
Welcome
Hana.
to our world.
Yeah.
Oh, this is a lot of sand.
Hana, you look cold back there.
What is that?
Frozen. And I brought more layers today, but I feel colder. I don't know how that works. I was like, I'm not going to be cold today.
Yes.
What?
Yes. Always. It's everyone's favorite candy. I'm a chocolate person.
Candy is nice for late nights.
Mm-hmm.
Burst of sugar energy.
Would
Yeah.
you be a hollow chocolate person or a solid chocolate person?
Ooh.
Mm.
Is this like a
Caramel
glass
filled?
half empty kind of thing?
Is there hollow chocolate
She
for
said she
dark
was a chocolate
chocolate?
person, that's why I was
Oh,
wondering.
dark chocolate's definitely
Yeah.
Sea salt
acceptable.
caramel
I'm hesitant
chocolate
to
person?
answer that one.
Because like
I don't
if
know.
it's dark chocolate, I
I don't know
will
how to
love
answer this. I like.
So.
I do enjoy
Like-
the crunch, but then I feel like you're missing out.
That's true.
It's like when
Also-
they imprint the,
What's this to the bottom
words
left?
into the chocolate.
Orange liquid
Words
orange
into it?
thing?
Oh,
Yeah.
here. I'll
Like-
line up. and
Yeah,
I'll
like
Can you get a
Hershey or
zoom in
anything
on
like
that?
that, and they print it in.
Yeah.
They're taking chocolate from me. Just keep it nice and level. I want every bit.
Every little inch of chocolate. No grooves, nothing, just the chocolate.
Yep. I opened the
What is
wrapper.
that?
I
Is
know
that
what,
a
I
dead
know what
piece
brand
of
it is.
coral or
I
something?
think that's just a.
I don't
Maybe
think
it's just so people can't
dead.
rebrand it.
Can you
Baby
zoom in
coral.
anymore? I don't know that it's a coral.
Just gonna angle it up a little.
I
if we
think these
wanna
are some
full zoom.
small still like stock algae around it. Or are those
Around it,
polyps?
but the orange part, what is it?
Well, you could take a representative sample.
You could.
We could. Can we zoom in anymore? I'm almost thinking
Enhance.
Where are
if
those
it's a
Thank you.
small round things with the white centers kind of all around?
That's an
they're
algae.
algae.
That's
Okay.
algae.
Yeah, that's
I think it's another
algae.
lichen.
Oh, what is that?
I wonder if this is It may be a soft coral, but
Is that a tiny little
I think
fish that
it's
was on it?
maybe it's
What is
it
it?
almost looks
Small
like a
fish.
sponge. The pink, see the pink?
Yeah, because you've got the
There's the,
oscula.
yeah, the little holes there.
Yeah. I don't know. There's not like a big exposed.
Yeah, it could
Osculum
be
at the
just like a
top.
small sponge. Did you get some grabs?
I did.
Awesome.
Neat.
Huh. Interesting. Hold on, pause for a sec. I'm trying to think if we should sample this guy.
Anything from science chat?
Well, there's no watches
No.
after this, so if you want to
This
fill up
is
those boxes.
true.
That's true. We're
How many?
coming up.
How many? What's our space like for sampling? We
Starboard BioBox, there's only one sample.
Okay. We haven't sampled too many. We have,
We got
let's see, 4 samples.
one moment.
samples.
Never let it go.
Oh man.
It's like a 8-mile quote.
Yeah, the water does look super like there's two different
Yeah. Oh, look at that
concentrations.
fish.
Hello.
That's a cool color.
Let's get a small snip of this guy,
Okay.
please.
Starboard BioBoxes are open except for A.
Okay.
And you got the stills, so.
Yes. This is going to be sample number 56.
Let's see. Depth 112.4. It might be a sponge. I think it's a sponge. I would like to get another. Let's see. We got a fan, a hard coral, a sea whip.
The random brown thing that we're not quite sure what it was.
Yeah, potentially a hard coral. And you said there was some small invertebrates, Peyton, that were sucked
in
up?
slurp chart number 2, while we were...
Any idea what they were? I didn't see them.
I didn't get a good look at them.
No worries. I just put inverts.
Yeah, I put some like I just wrote random inverts, but like
Okay.
in the notes it's like light attracted invertebrates.
Okay.
I would guess mysid shrimp, but it'll be neat to take a closer look once you're at the surface.
It's yeah, that would be a good guess.
Do you want to half zoom?
Sure. Half zoom.
This is going to be tricky.
Yeah.
Oh, it looks really small. So
It is pretty
maybe just
small.
the whole thing. How big is it?
Just that clear
Oh.
thing. That just...
Oh, maybe that part on the right, you could, I think it might be separated, and you could,
Hey, do you see that kind of ribbony looking thing?
Yeah, that's probably a worm.
Oh yeah, look at that.
Hey, bud.
So clear.
Mm-hmm.
I don't know if you could angle the arm. Let's see, how would you do this? With the claw facing to the top left corner, so then you could just snip the one nub to the right without clipping the others. I don't know if that's possible.
The blades are in the back part, so it's like
yeah.
you'd kind of crush it.
I see, okay. The claw.
Go, Scott, go.
Oh no,
It's moving the whole thing.
Definitely seems spongy.
Yeah, I think it's a sponge.
Oh.
Whoa. You think you could cut that piece off?
Trying.
I don't know if it's going to separate.
Without using the cutter, that is.
It's very flat now.
Yeah.
Should we just take the whole rock?
It looks like those are two sections.
Yeah, it's almost like maybe we could split. Oh, look in there.
Oh. Do you think you can cut that section right there between them?
Go, Scott, go.
Maybe you could cut in between it now. It's vertical. Patience. Oh.
You got the whole thing.
Did we not get it in the...
It didn't cut.
Not cooperating sponge.
You had one job. And this...
You can kind of see it, right? It's not cutting though.
Yeah, you can
The
see it in
sponge
that
had
clear
one
section.
job,
Yeah.
and that was to cut cleanly when we took an ROV claw to it. And that's with the, like, it's... Oh,
Nope.
Oh.
that is a resilient sponge.
Yeah, pretty hefty.
What if it just, like, bent the coral cutters?
I don't think it's that hard. That would be pretty crazy if it did. Tricky little guy.
Ooh.
Ooh.
Do you get anything?
I don't think so. Nope.
Where'd it go?
Yeah, where did it go?
It
You want to sample
did
the whole
snip
thing?
off there, didn't it?
Not
I don't
particularly.
think
It's
so.
Yeah.
slipped through.
I thought it was in there.
Maybe try to prop it upright again. Might be easier.
I mean,
It looks loose.
Oh, there's like a little piece dangling.
Yeah.
It's just small.
I mean, at this point, is it going to be able to continue on?
It's
It
almost
will.
cut.
Oh, yeah, yeah.
Oh, I see it, yeah. Yeah, sponges are modular, so they can definitely regenerate just fine. You can cut a sponge and then into a few different pieces and stick it in various places and they'll regrow. One really cool thing, like when you think about corals, in a way, they're immortal because they don't age. So as long as they don't get a disease or have any like hard physical damage that causes them to die, or bleaching or other stressors, they can technically keep growing and don't have per se like an expiration end of life date because they don't age. So like Big Mama could be growing for 500 more years. Definitely can happen. And then one nice thing about corals, the bigger they get, their fecundity output increases. And it's always good when you have large corals like that because they put a lot of gametes into the water column, and they're obviously resilient because they've survived so long. So when they crossbreed with younger corals, it adds more resiliency to the future baby corals.
That's pretty crazy to think about. Immortal.
Yeah, because there's, I mean, most corals these days are dying from like anthropogenic influences, you know, runoff pollution. And from that, they're catching diseases, or the more recent global warming and mass bleachings and stuff like that. But if they don't have any stressors, oh my gosh, this guy will not.
This is.
It's like barely hanging on.
Wow. Oh.
Oh man.
The sponge is.
Are you gonna let us just take a little piece of your nub?
What'd you do today, honey? Nothing.
Harassed a little coral nub.
I fought with a sponge.
Yeah, literally. Oh man.
Because the glass sponges, it's like one hit and you're already
Just like
cutting them. Yeah.
These ones are more like, I guess, rubbery in a sense.
I would say it's definitely immortal.
Do sponges age?
No, I believe they don't either. So I know like in the Caribbean we have these big barrel sponges, and they call them the redwoods of the deep sea because they can get exceptionally large. And same, they're modular organisms, so technically they can grow until they an
That rock
outside
is not in the right spot for you.
factor kills them.
Yeah, get out of here.
We're redecorating.
Move.
Will the world end if we just take the whole thing?
I know, I'm debating to, I'd like to not take the whole thing, but I
don't know.
to.
Yeah.
Scott's got this.
So Scott, are you using the little like replica arm to control the actual predator arm?
Yes.
Oh,
Mini master.
that's
Gotcha.
pretty cool.
How difficult is it to control using like the little arm?
Extremely.
Exhibit A. It depends too on the types of draws you have on the end. Like these are kind of harder, I think, because they're the coral cutters.
Yeah.
But when you have like smaller fingers, you can grab tinier
It's easier.
things, and
Yeah.
they're kind of easier to like maneuver, grab stuff.
You got it. Nice.
Our viewers are in support of just taking the whole thing.
Might have got it.
Yes!
Oh,
Yes!
Woo! Success.
All right.
Oops, sorry. Oops.
Oh, I see the little piece. Awesome.
We're putting it in a box.
Okay.
correct?
Preferably starboard bio boxes.
Yeah, we're ready. Which ones?
B, E, C, D, F are all open. So whichever is easiest for you,
Mind if I
Scott.
zoom in on Argus?
Yeah, go for it.
Sorry, what was your question?
Ready?
Ready.
We actually are like, from where we started, we are
So B, C, D
directly
are the last
south.
3, and then I don't.
From our very
E.
initial where we
You said
started on
E?
our watch. Yeah, that's where we started on our watch.
A-
So we
Everything
went,
is open except for A.
we went
Everything but
east
A.
and came
Okay,
back
so
west.
any of them, yeah, except for that back one.
Yes.
Or this front one, sorry.
So you
So,
guys are
like-
welcome to do any container that's easiest for you if you're worried about it drifting away.
Yeah. You want to get to the point?
Nice.
Yeah. On the ridge. Well, we only moved 20 meters too. So it didn't move very fast that far.
So let's, yeah, we'll keep going some more and hopefully.
All right.
Awesome. Thank you, Scott.
You're welcome.
Thank you, Scott.
That's tip jar is behind me.
I think that was the hardest sample we've had to get.
Yeah, that one was a little
So,
Uncooperative.
uncooperative. Yeah.
yes. Question, we didn't have start or end time for the photo mosaic, did we? The one we did? I forgot to write that down.
I think we do.
Okay. If you have it, that's fine.
Yeah, give me 1 second.
A lot of algae.
Let me just clean
Summer's
up my
turn.
browser.
Go Summer!
Woo.
Did you have a start time? I kind of have a start time on that, Peyton. If
I can pull it up.
okay.
Let's see.
You're full light on both.
Well, I don't need it. Just
That
if
was PM. So,
PM1.
yeah, 12:25 was our
Yes.
start because that's when we started doing the test.
Okay.
And then it eventually turned into the photo mosaic.
yeah.
Yeah.
And then we ended our photo mosaic at,
Good l-
is this the end time? At 12:38.
It's gonna say.
12:30. Okay, awesome.
Can
Thank
you head towards 140? Yep, you're perfect.
That was some serious ROV skill.
Yeah. Took a while.
Let me. How much?
Next time we should just have Bobby Argus do it.
Where is it?
Yeah, he's down
Got
there
it.
already.
Be easier.
Okay. 15 to 18.
Yeah, it's a 20-meter step, so it's a pretty big step. I think he's gonna add another.
Get to something more interesting.
You're at 15. Yeah. That fish came
Whoa.
out of the ROV.
Yeah. Ching.
Okay. Are you still coming up?
There's a couple fish down there at the
Right.
bottom, right hand corner.
Yep.
And one
The ship's gonna
Oh,
settle
there's
out
a
right
bottom
here.
left.
Yeah.
Okay. Do you know what time we need to start ascending? Because I know we need to be...
We're
So,
really
like,
shallow.
So we
We're at
probably
100
have like
meters.
about an hour left, maybe a
Ship's
little
settling up.
less.
Yeah.
Okay.
It's at 18.
Yeah, I mean, it's going to take us like 10 minutes to come up.
Okay.
Well, like 30 minutes maybe.
Yeah. So around
thank
3:30,
you.
we'll have to
I am still coming up.
Yep.
This is just
call
like a
it
crouch
quits.
hold.
I'm watching it.
Cool.
So Hannah, we have a request from a viewer to kind of explain more about what you mean by corals don't age. That's a kind of crazy thing to think about.
Okay, slowing down.
Yeah, so
I'm gonna take another step.
let's see, how's
yeah,
the best
we're
way
not
to
gonna do stuff here.
explain this? Give me 1 sec, I need to take a note.
Mm-hmm.
What is done?
Hannah, we're just gonna keep going because there's not.
Yeah,
unless you wanna
let's,
stop
yeah,
or-
exactly.
okay.
Let's just keep going
Keep
until we
on.
see something of interest again.
Roger.
Yeah, there's not a whole lot here. I'm almost wondering if we go up and maybe go... Oh, there's a trevally.
So we've gone about 40 meters since we first started going up the hill. So I figured at the end of this little step that he's doing, we can start heading back east if you want to.
Yeah, that's where
go
we
back
saw, I
east.
think, the more interesting stuff. So yeah, that sounds like a good plan.
Okay. So once the
And
ship settles out again.
I'm kind of debating too if we should go all the way up to T4 and then cruise laterally, like kind of just before it maybe, with the thought that we'd see more there. But let's just keep continuing
T4
up
down
and
here.
Okay.
Oh.
Yeah.
we'll kind of see what we see.
Those long silvery fish are trekking down there.
How did that happen?
It's a good sized trevally.
So we've gone about 50 meters. Do we want to go across or do we want to keep going up?
Maybe, let's see, it doesn't look like there's much. Let's maybe go like 20 meters to the east and just kind of see, and then it doesn't look like there's much, but just to kind of scope it out a little bit.
Alrighty.
Okay.
I can think with 19 cool.
We kind of have to keep still going. Yeah, the slope's pretty much east, so. Well, I mean, it's to my left, so it's like southeast still, so you still have to go southeast, right? You just keep this bearing. One. Oh, more east?
Yeah, just so we can hopefully see more, just because there's not much to see here.
Okay, yeah, that's fine. Yeah, that's fine.
Yeah, it's just a
Oh,
bunch
I
of
kinda-
sand.
If we go 100.
So I'm looking at a paper actually. Someone did a research paper about do reef corals age?
Mm-hmm.
There's like a sand channel. That's interesting.
I'll take a note of that about it.
So yeah, so back to modularity. So it's refers to the ability of an organism. So if it's cut, it can regenerate, and it'll, I guess you could say, like seal that part of the tissue. And then it can also translocate nutrients too, so that helps in keeping it alive. But they don't.
More south step.
I'm trying to remember
Okay.
Like, let's see, how can I explain this?
She wanted to go, yeah. You got a little ahead of the ship. I'm gonna let it finish this, not finish, go a little further, and then adjust it. So.
I want to say they're,
yeah, I'm just gonna keep going 140.
like, they don't have telomeres like we do, so they can't age.
Mm-hmm.
But they can also, because they can translocate and asexually reproduce, and they don't.
Can I jump in?
Yeah, please do.
Okay. So it looks like there are a couple species of corals that are considered short-lived. And
Are those deep sea corals?
no, I think these are all like
Just
probably
in general.
shallower. So Stylophora pistillata is cited as like a shallow, like short-lived species, but a lot of the longer species, like I'm reading this paper right now and it's talking about like trying to look at multiple areas where corals may be aging. Like they do find that corals do generate mutations over time, but that their gametes don't have those same mutations. So it's a somatic versus like somatic versus sex cell lines, which is really interesting. Like there's a separation of them. And then it does, I think it briefly talked about telomeres. Let me see if I can find it in the document again.
Yeah, let's see. So I guess when you think of it in a sense, like to be immortal, like Peyton was just saying, that the gamete, so it can continuously reproduce over time, and that's, if it reproduces asexually, it's a direct replicate. So if a coral breaks, and then you have ramets of that coral that fall down below, it'll continue to propagate asexually. And so if one of those chunks dies, technically still those other remnants that were from, say, the mother colony continue to propagate and live, and or all of the ramets continue to live. Unless there is an outside stressor that ends up killing them. And then they can continuously asexually reproduce and get to larger and larger and larger size.
Yeah. Okay, so what I'm reading here is that different corals may behave differently when it comes to telomeres and telomerase. So some corals have much lower, so quick science lesson. So at the end of your genome or the end of your strand of DNA, there's a section known as the telomere, which is basically like kind of throwaway DNA. It's extra nucleotides that are added on to prevent.
Code for anything.
Yeah, it doesn't code for anything, and it's added on to prevent. Basically, like each time your DNA gets copied and a cell reproduces, you lose a little bit of your DNA. That's why the telomere, so that section of DNA, shortens over time.
Nav, can I get a zoom out on the map, please?
So there's
I feel like there's even less now.
there's an
And
enzyme. Oh, sorry. Go for it.
Yeah. Let's see. I was really hoping we were going to see more.
Yeah. Oh, there's a big coral on the left.
On the left.
Yeah.
The kind we sampled, right?
Oh,
maybe.
let's zoom in on that.
Yeah.
There's a fish.
Well, a couple fish just went underneath it. That's funny.
Nope.
There's some fish over there.
Oh.
We got fish.
Oh, there's a
There.
couple
Let's
right
check
somewhere on
out.
the left.
Yeah,
Oh,
There's
we
some
found
coral.
the place.
Is it
let's
another
go to the
unicorn?
left and check this out.
Oh.
Look at all the fish underneath that.
Are those
Oh,
angel
Not the Argus
fish?
hand.
We
It's like
found
little
a spot.
patch.
Yeah.
Photo mosaic.
I was just thinking that, really, I think we should do one right here.
Yeah.
Look at the fish. The coral.
Very cool.
Oh,
On a side
Ooh.
note,
We turned that off.
bannerfish.
I turned off.
Look at
bucket
all the
cam
fish that
and
are right
rail
down
cam,
there.
And the DC ground faults are up to 2 now.
What are those red, sorry, black-white fishes?
I'm gonna keep
Those are
keep
bannerfish, I
cameras.
believe.
um,
They're like
That's
similar in
unicorn
pattern
fish.
to
Chromin
marsh
bottle on?
idols, but they're a little bit different. Oh, and
I
the
will do
trevelies
that.
are here.
Just
Oh,
for you.
here's a bunch
thank
of trouble.
you.
What is that?
Look
a
at all the
cowfish
fish.
right
Oh
there?
my gosh, that's a
Yeah, I
cowfish.
was
Can
just
we get a
seeing
picture of the cowfish, please?
Where's the cowfish?
There's
It's
a
Straight-
unicorn fish
like
and a cowfish.
straight center. It's
Yeah, yeah.
like
Six
the
o'clock.
The
boxy
greenish
fish.
one by
next
all the
to
red
the
ones.
red.
Are
yeah.
those
Yeah.
squirrel fish?
Mhm.
It's that one. It's this
Sorry.
one. It's that one. Go, go,
I'm,
go.
I'm full wide.
Hurry.
That one.
So much going on.
Look at that
How many
fish.
voices do you need in your head?
They're super cool to check out.
Sorry, Scott.
Okay, let's see.
90s.
We all should start talking.
There's so
It's
many of those
exciting.
little
It's-
squirrel
it is.
fish.
This
I
is
hope,
a great
I hope
little.
that our viewers are also equally as excited.
About our little catfish.
Check out this cowfish.
Look at
Oh
it
my
all.
Dump my
goodness,
coffee,
I
I'm ready to
love
go.
it.
Woo!
Okay.
It's so good, you guys.
I was gonna say apparently their meat tastes really good.
Oh.
What?
Catch
cow-
him. You
it's
can
the-
grab
Wow,
him right
he's a
now.
pretty fish.
Reach through your screen.
Go to sleep.
Wow, cool little spot
You're done
pattern.
here. You did a good job. You're dismissed.
Oh, no, no, no. I got 2 more hours. Or I guess 1, sorry, not 2.
Maybe like 40 minutes-ish.
It's a cool cowfish.
Yeah.
Look at all these
La,
fish.
la, la.
Look at all these chickens.
Funny looking
heck?
mouth.
I'm turning cameras off to suss out this.
Oh.
DC
Oh,
grams.
to finish up what I was saying earlier about
You wanna
telomeres.
look
Remind
at
Basically-
me, Summer, we went east,
Oh.
correct?
we went southeast.
Southeast.
Yeah,
good.
we couldn't
No
go
time for-
exactly
no
east
time for
because of
the
the slope.
science.
We're good.
No worries. I wonder if that's why we saw some more stuff.
What else do we want to look
What are all
at?
those red fish?
I'd like
I
to
think
do
they're
a
squirrel
photo
fish.
mosaic grid here.
I'm
Okay.
setting up the cameras. We
just
had
call
to
them- Oh, okay.
reboot them all. Yeah,
that's
so
Yeah,
give me a
they're
few
back.
minutes.
They're back
No
on.
worries.
Enjoy this reef that we're
Yeah,
looking
let's
at.
keep zooming
Oh,
on
they're
stuff
not
until she's ready.
Sorry, they're not squirrel
Well,
fish.
those are
They're
big
something
fish.
else. Hold on.
They
Look at all
look
of
like
them. There's
squirrel.
so
Actually,
many.
no. Big eye squirrel.
Yeah, so they look just
You
like
want to
them.
zoom?
Maybe. Okay, yeah, sorry, they are squirrel fish.
They're so cute.
There's so many.
Okay, I want to finish what I was saying about telomeres.
Oh, do you see this tiny little fish? He just
Where?
Which
snuck-
ones?
He was at the bottom, about 6 o'clock. He's towards the center of the screen now, but he just went behind little corals.
These fish have the perfect place. to, like,
Describe
chill.
it better, Brian.
He was
Can you get some
tubular in
stills of
nature.
this?
I am definitely getting some stills of
Awesome.
this.
Are the lasers on? Yes, they are.
Is there anywhere you want to look specifically, Hannah?
No, I just want to get an overall
Do you see, what is that?
capture.
Eel
Thing
over
moving
on the
around.
left.
Oh,
Yeah, the gray, like, sort of flapping.
yeah. It
Oh,
Oh,
might be a
it's coming
trigger.
out.
Might be a type of triggerfish. Come
Right,
out.
go ahead,
Is
video.
there a
Oh, is there a second
Is
one
there a
up
second
top?
Oh,
Yeah,
I was,
there's-
yeah.
No, no,
there's
you-
Do you
two.
guys see
Oh, there
the
is two.
shape, like, wedged inside
We're
of
looking
the...
at different
I
ones.
need to concentrate.
There's two of them though. Do you see? There's one in the middle of the screen and then one in the
Oh.
upper left.
Yeah. Can you look at the one on the upper left? I
Put
think
the
we
arm
can
in
see
there.
it better.
Scare it out.
They're totally
Yeah, I
scared.
can
They're
look at
gonna
the one
like, they're not
Yeah,
gonna
okay.
move.
Did you notice how they were moving by their fins? Oh, yeah, they are
Kind of s-
triggerfish.
Yeah, I'm pretty sure they're
Yes.
triggers.
I can see the
Here,
fin.
wait,
Oh,
zoom
zoom
out,
out.
zoom out. I'm gonna look at the other one. Yeah,
Yep.
this is a, yeah,
Pretty real.
so much fun. I love hearing all your voices at the same time.
Can you
Not
get
just
another
one
star?
at all.
Some
Yep.
real life,
I've been grabbing them continuously.
It's just a nice, pretty.
Train.
Oh, is there
Oh,
another
for
one?
sure.
Is he hiding over there?
Is there two? No, there's
Yeah,
only one.
yeah, no, there.
There's one and there's
There's-
another
this
one
is the
closer
second
to
one.
us. And they're both very freaked out.
There's one there,
Yeah.
and then
Oh, there was one
Oh,
He
down
went,
there.
There's a big guy in the back. Look
he
at,
hid.
look out.
Wow, that's really pretty.
For sure.
Yeah, we're
Can we do a fish transect right here?
2.1.
We're gonna do a mosaic first.
Do you want anywhere in particular to start, or like, I mean, this area is.
How are we going to count all of these
Could
fish?
you maybe like back up or come off the box? Like right where we're, like what's right in front of us would be nice to capture, but maybe I want to see like behind us. I think there was a little more, so maybe we start.
If you
a
look
little
on the
behind us.
Argus view, it doesn't look like there is actually
Oh,
too
I
much
see.
behind us. It looks
Oh,
like we
no,
just.
Is that
Okay,
another?
so
Is
let's,
that a parrotfish? right there in front of us?
I don't know.
It almost looks like a cross between a parrot and a tang.
Yeah,
Yeah.
it does.
That's an interesting. I've never
It's
I
seen.
a
think it's a unicorn fish. Do you see the two spots on the
He's
tail?
got the spines.
No, I.
It's got the two blades.
Oh yeah, it does.
This is totally Monterey Bay Aquarium stuff going on right now.
Oh, yeah.
Okay, so let's maybe.
You didn't turn off the bottle, did you?
No. No, I definitely did not do anything
Okay.
with the bottle.
I
Sorry, let me
definitely
prep
like
for
from
the-
the right
cycle
to
it?
the left, kind of
No,
get
no,
this
no,
plot
no.
of coral.
Alright.
I don't know how
So,
big of a plot that
Look
would
at the
be.
left. Oh, start, you mean to the left?
Yeah, I don't know how big of a plot that would be.
What kind of
Let's
coral is
see.
the one on the left?
I
Well, it's now
lost
more front.
corals.
The like
I'm gonna
bowl-shaped
say-
one.
Yeah, the bowl one.
I think it's just called a plate coral.
10 by 5?
I think.
Is that what that would be? 10 by 5?
It's probably 10 by... Right, 10 by 10. I was just
yeah,
thinking...
10 by 10.
Okay.
They're coming to investigate.
And I
Um,
would, yeah, so you're thinking
Oh,
about
those banner fish
sliding
are very
over to
pretty.
the left.
Yeah,
and
to
start
get all
there.
of it. Yeah.
Yeah,
Oh,
Agreed. There's
yeah.
nothing behind you.
Oh, there's
Start right
another
here, just-
something popping out there.
But to the left.
What is that?
Yeah. So bubble starts out up. So you
Oh.
just
Another
bring it-
unicorn
yeah,
fish.
Another
Just
unicorn
bring it-
fish.
Yeah, or you,
Hello, friend.
and it does that
so
too.
you think a 10 by 10 would encompass
Just
that
hit
whole
the
frame?
joystick and it like
That's a good question.
do
10
it
by
again.
10 will, maybe 10 by 15.
Okay, that might be too big. So let's
Yeah.
try to do 10 by 10 kind of in the most concentrated area.
Okay, well, maybe like 2 meters to the left to start, and then
Yeah.
That looks good.
and then 10 forward would get us past that large white coral. I don't know the name of it.
Oh, the soft coral straight ahead,
Yeah.
almost dead center. Yeah, okay.
And then we could just, at that point, we could just keep going as
Oh,
far
is it?
right as you want to go.
oh, it's like fish.
Okay.
Is that the
Right.
same
Yeah,
fish we
that
saw
sounds good.
before?
Start
I
about a
think
meter
so.
to the left then.
Yeah,
Yeah, so that way we get these hard corals that are
Yeah.
right in front of us.
yeah. We've got good Doppler, so this will be easy,
Awesome.
easy
Alright,
pickings.
it's just-
So Peyton, how's counting all the red fish going?
Do you wanna
Get a couple more
zoom in on
still.
stuff, specific
Yeah.
stuff?
I don't know. I
this is
do.
incredible.
Yeah.
Yeah, we'll do.
Anthony, there's at least 5.
Okay. I think that's true.
Okay, so this would be photo mosaic
I'm
2.
not
Count
wrong.
10. I count 10 so far.
Look how
there's
long
another
they're-
111.
the fish's the doohickey is. You know,
They're-
that's not a
they're
technical term.
Top fins?
Yeah, what's that? Is that actually a fin?
All right,
Yep.
bottles are back on.
Really?
Yeah, Moorish idols and bannerfish both have that fin. I actually haven't heard a good explanation from any biologists about why they have it. I think, like, my personal guess is that it's an indicator of, like, age or, like, you know, fish that have that intact might be able to breed more effectively. I'm not sure. Like, because it's, like, a symbol of, like, status and survivorship.
Huh.
But males and females both have it, correct?
Yeah, I think so. So I would not know.
My guess would be, yeah, be related to
Yeah.
mating,
It
but
was
I don't know if both sexes have it or not.
I think they do. I'm pretty sure.
Mm-hmm.
But yeah, I've never had a good explanation from a biologist, so that's my own personal guess. Oh, I'll get some crabs with these guys.
Is that close up enough for you?
Oh, oh.
I need closer.
Can you enhance? I need to see the atomic level.
That's the most zoom you got.
It's not acceptable, Anthony. I need greater detail.
What do you mean there isn't a scanning electron microscope
know,
on the
right?
ROVs?
I'm so sorry, Peyton.
I'm disappointed.
I'll add that to the list.
There's some
Before or
sponges to
after the
out.
masseuse?
I like all that it looks like lichens everywhere.
Yeah, it does. There's a
Well,
lot of
hi,
like
fish.
encrusting.
Welcome back.
Such a pretty scene.
I'm just blown away just by all the different fish we're seeing. It's just,
So many colors.
yeah.
I'm surprised that they're so comfortable getting this close to the ROV.
We're part of the family now.
Oh,
We
look at
really
that.
did scare that triggerfish.
Oh yeah, I wonder.
The one that
There's-
was over here is gone, but the other
Oh, did one
might.
of them leave?
There was one like right in this cavern right here, like totally freaked out.
It
and
jammed
then-
itself further deep.
Yeah.
It's so funny. When I've snorkeled, I've seen them, and they go under a rock like that. And one time there was one, he was under a rock, but it was like barely under a rock, so I could completely see him, and he was totally freaked